Hey everyone, and welcome to this definitive test prep explainer on biomolecules. Today, we're unpacking exactly how non-living atoms self-assemble into incredibly complex living systems. So if you've ever wondered how the food in your grocery cart, or maybe that healthy lunch sandwich you're about to eat, magically becomes a living, breathing organism, well, you're in the right place. We're going to break down the biochemical logic of life step by step so you are absolutely ready to crush your exam.
Take a look at this incredible quote. It really sets the stakes for our study session today because it reminds us that life isn't magic, it's just perfectly synchronized chemistry. When you strip away the mystery of biology, what you're left with is the domain of biochemistry. It's the study of how structural biomolecules interact to sustain, grow, and reproduce a living system. Literally, every breath you take and every thought you have is driven by this harmonious progress of chemical reactions.
Here is our roadmap for today. We'll cover 1. The toolkit of life, 2. Carbohydrates, the energy spectrum, 3. Proteins, the biological machinery, 4. Regulators, enzymes and vitamins, 5. Nucleic acids, information code, and 6. Synthesis of living systems.
All right, jumping right into section 1, the toolkit of life, 4 modular architectures. I want you to burn this visual roadmap into your memory. Think of these as your 4 core tools. First, you've got carbohydrates for energy. These are your body's fuel cells, kind of like the bread in your lunch sandwich. Second, proteins, the physical machinery and architecture, like a slice of turkey. Third, nucleic acids, the biological software. And finally, enzymes and vitamins, the essential catalysts making digestion and growth possible. Everything we discussed today is going to fall into one of these 4 buckets.
Moving on to section 2. Carbohydrates, the energy spectrum.
Think of D-glucose as your absolute primary energy monomer with the chemical formula C6H12O6. If you're looking at a nutrition label in the grocery store, this is the fundamental sugar. Now, in the real world, we extract this via hydrolysis. You can get it from cane sugar, sucrose, by boiling it with dilute acids, or commercially from starchy foods like potatoes or wheat. Glucose is literally the fundamental battery pack that fuels your daily physical activity. Now, for the exam, you have to be able to contrast simple base units against complex networks. Monosaccharides are the base units. They cannot be broken down by water into anything simpler. They're your quick-hit, processed sugar energy spikes. On the flip side, you have polysaccharides. These are massive networks that yield huge numbers of monosaccharides upon hydrolysis. They usually aren't sweet, and they provide that sustained, long-term energy release that is absolutely crucial for athletic endurance and a healthy, balanced diet.
Notice how those stage 1 base units actually link up to form vast stage 3 architectures. Starch acts as plant storage, cellulose consists of rigid units forming plant cell walls, and finally, glycogen, that highly branched structure you see there, is animal storage. So when you're playing a sport and your body needs a sudden burst of energy, enzymes will rapidly break down those complex glycogen branches right back into simple glucose fuel. Pretty cool, right?
Okay, let's switch gears to section 3, proteins, the biological machinery.
Proteins are built from an incredible alphabet of amino acids. Every alpha amino acid has an acidic carboxyl group and a basic amino group attached to a central carbon. You can think of this as a chemical two-way switch, the Zwitterion toggle. In step 1, the carboxyl group loses a proton, and in step 2, the amino group accepts it. This forms a neutral dipolar ion. It's amphoteric, which just means it can react with both acids and bases. And honestly, this flexibility is the entire secret to protein versatility.
Make sure you memorize this 3-step polymerization process where these amino acid building blocks forge peptide bonds. Step 1, you align the carboxyl group of one amino acid with the amino group of the second. Step 2, dehydration synthesis occurs. You literally eliminate a water molecule and H2O from the two groups. In step 3, they snap together, forming a peptide linkage. Chain more than 100 of these together, and boom, you've got yourself a massive protein molecule.
Now, this sequence of amino acids creates a fundamental 1D primary structure. But proteins don't just stay as flat chains. They fold up this staircase into specific 3D molecular shape archetypes. Hydrogen bonding creates secondary structures like the twisted alpha helix or the draped beta-pleated sheet. Further folding, stabilized by disulfide bonds and electrostatic forces, gives you the tertiary structure. Ultimately, you get highly specific shapes, either fibrous proteins like keratin that are parallel and insoluble, or globular proteins like insulin that are coiled, spherical, and soluble in water.
But remember this crucial real-world application for your test: denaturation. A protein found in a biological system with a unique 3D structure is in its native state. But if you apply a physical change, like heating, or a chemical change, like altering the pH, you unravel that 3D structure. Think of cooking an egg white or milk curdling in your fridge. Denaturation completely destroys biological activity. But, and this is key, the primary structure, that 1D sequence of amino acids, remains perfectly intact. Only the secondary and tertiary structures are destroyed.
Section 4 brings us to the regulators, enzymes, and vitamins.
You've probably heard this one before, but definitely anchor it to the classic lock and key analogy. In biochemistry, you have a substrate, which is the specific molecule being acted upon. Think of the substrate as a highly specific key. That key fits perfectly into the lock, which is the enzyme's active site. Because enzymes are highly specific globular proteins, they will only catalyze reactions for the exact substrate that fits their active site shape.
Enzymes are doing the heavy lifting by sequencing biological reactions under mild body conditions. But we also absolutely rely on vitamins. These are essential organic compounds our bodies simply cannot synthesize on their own. We have to get them from our diet. For example, vitamin E, sourced from grocery items like sunflower oil or wheat germ, is needed to prevent increased fragility of red blood cells and muscular weakness. Or vitamin K, which you get from green leafy vegetables and is completely essential for proper blood clotting.
Let's dive into Section 5, Nucleic Acids, Information Code.
So, what's acting as the architectural blueprint programming all of this complexity? Nucleic acids. The fundamental monomer here is the nucleotide, and it consists of 3 components. First, a central pentose sugar hub, either ribose for RNA or deoxyribose for DNA. Second, attached to the 1' position of that sugar is a nitrogenous base, like adenine, guanine, cytosine, or thymine. And third, attached to the 5' position is a phosphoric acid. Combine those 3 and you get the fully assembled unit, the nucleotide, the very building block of heredity.
These individual nucleotides link together via phosphodiester linkages between the 5' and 3' carbon atoms of the sugars to form a primary backbone. But here is the absolute genius of nature. 2 of these chains wind around each other to form the iconic double helix. The strands are complementary, held tightly together by specific hydrogen bonds. Adenine always pairs with thymine, and cytosine always pairs with guanine. This elegant, unalterable sequence is exactly what dictates your unique biological identity.
All right, let's bring it all together for the grand finale in Section 6, Synthesis of Living Systems.
Remember, a living system is composed entirely of non-living atoms and molecules, but life itself is the emergent property of these tools working in perfect harmony. Just look at this cycle. Nucleic acids act as the reserve of genetic information and direct the synthesis of proteins. Proteins then provide the physical architecture and form enzymes. Enzymes are the biocatalysts that coordinate the breakdown of carbohydrates, and carbohydrates provide the vital energy required to power the entire cellular machinery, allowing nucleic acids to replicate, and the cycle continues beautifully.
Before you head into your exam, let's do a really quick practice quiz. Which modular architecture acts as the biological software, and which acts as the fuel cell? Think about it. If you answered nucleic acids and carbohydrates, you are absolutely ready. As you review, ponder this overarching truth. If life is simply the perfect harmony of these biomolecules acting together, what reaction will you catalyze next? You've got the toolkit. Now go crush your test. Thanks for joining us for this explainer.